Before we move in, I'm afraid you're going to have to work for your supper tonight. There's a little bit of a tutorial, which we're going to talk about. And my colleagues, who I'm going to introduce in a minute. So I've got Dr. Alex Webb, who's a senior research associate in my lab. Kei-Ann Wen, who's a fourth-year PhD student in my lab. And Dr. Kirsten Jensen, who's a senior research associate in our group. And they're going to just set off and do experiments, so ignore them, please, if you can. But they're just going to do some stuff, get some stuff going, and then we're going to come back to it at the end. But I want to take you through a little bit of a tutorial to start off with. And I think this all concerns this DNA molecule. I mean, this seems to be an incredibly important molecule. You know, why is it so important? So I just want to run through that a little bit. And I think because DNA encodes pretty much the instructions for all living systems on planet Earth. And I ideally hope that before I leave this planet that we find something like DNA on Mars, and that would be an extraordinary discovery. So DNA is sort of like an operating system. If you can imagine the operating system on your computer, I've got a Mac, so I use OS Yosemite. It sort of provides all the instructions for living systems. So it provides everything that you need for a living cell, and therefore it can be passed on to subsequent generations, and it is essentially the essence, if you like, of living systems. So I guess the big question is, you know, what is it? Okay, so it's a very simple chain of 4 chemical building blocks, G, C, A, T. I won't necessarily give them their chemical names. They're up there if you can see them. And then it forms this lovely kind of pairing. So you'll see here that the red will only go with the green, and that the blue will only go with this sort of orange color. So you have this G will always go with C, and A will always go with T. And then it folds into this beautiful double helix, which everyone understands. So this essentially is the instruction set for living systems. Now, if you were to take all your DNA out of each cell in your body, it would stretch to the moon and back 6,000 times. So that's one individual. It's an extraordinarily compact, information-rich molecule, and it's at the center of every living system. Another aspect is that 1 million bases of DNA encodes about 1 megabyte of computer data storage. You've got 3 million bases in each cell, and you've got millions of cells. So you can imagine you as an individual, at least your DNA, is an extraordinarily rich storage material. Now, it's a complicated, it's a simple molecule in the sense GCAT, but it's complicated when you have millions of these chains linked together. So in computer terms, we usually refer to it like this. We have a sequence, a DNA sequence, GCAT, GCA, and that's the sequence. Now, so the question is, how is the code, which is the instruction set for all living systems, translated? So how do we translate it? And what information does it store for living systems? So this is now the central dogma of molecular biology, and I won't spend too much time on this, but there are 2 steps that you have to understand because this is the basis of synthetic biology. That is that the DNA is changed into a molecule called RNA, and then that molecule, messenger RNA, is then translated into proteins. So DNA codes for all the proteins that we have in our cells. And therefore, it makes all of our cells operate and work because it codes for the master molecules, I think, which are the proteins. Now, you'll see there's a machine, there's a polymerase. It will copy one chain, and then that chain will go into this machine called the ribosome. And the ribosome then dictates how you link the different amino acids that form the protein. The point here is that the protein code is made of 20 amino acids. Now, if you lift up the cards showing the single letters, and I know there are a few people absent, but you in this room, and I think the camera's going to click in and show you, hopefully, what you're seeing, I believe, in a second. Great, there we are. There's a picture of the selfie of the audience at the Ellison-Cliffe Lecture. What a brilliant shot. You have got a lot of GCAT, GCATs. Now, what are you coding? So if we go back to the PowerPoint presentation, in this audience, we've managed to find 297 cards, and you are coding this genetic sequence. And this sequence codes for a protein called HIV-1 protease. Now, that is a pretty important protein, particularly if you're treating HIV. So that is a genetic code in this room. So now the question is, if you reverse these cards, and you'll notice that only every 3rd person has actually something on the back of their cards. And if we flip through there, you'll see that there's a whole series of 3-letter codes, and that codes for the amino acids or the proteins. So we've now translated our genetic code into a whole series of linked amino acids. And if we go back to the PowerPoint presentation, you'll see that that is the code that we've got. So in this room, you have the genetic code, and then it's reduced down to the protein code or the amino acid code. And then finally, this amino acid code folds up into a three-dimensional structure, and there is the protein structure of HIV protease. And there are two proteins, two monomers come together to form this intimate dimer. And in the middle of that movie, you'll see a little blue blob, and that's where all the retroviral HIV treatments, therapeutics, are targeting. So the intimate shape of that molecule allows you to target all of the interesting pharmaceutical molecules that have essentially had a huge impact on the treatment of AIDS, as you can see from this particular graph. This is just on the side. So therefore, I chose that example because I think it's incredibly important. And so the HIV protease inhibitors, as you probably know, medics will know, has been incredibly influential in essentially reducing death rates in HIV. Okay, so what does the DNA look like? So a lot of people think, so we've coded it, we've kind of now translated it into a protein, and I've shown you what the protein looks like in 3 dimensions. Now we want to look at what the DNA looks like. Now here I turn to the faithful strawberry, because the reason I choose strawberries is that strawberries have got a lot of DNA. Now Kirsten's going to show that now, so if we just show what it looks like. So Kirsten did this experiment where she put some, mashed up some strawberries, put some pineapple juice in, and then put some alcohol in. And in a minute, you'll be able to see what the DNA looks like, the real DNA from strawberries. And it is all of that lovely white stuff. And you can start seeing some strands coming down here. I don't know if you can see those, but you'll see some little sort of strands. So all of that is essentially DNA. And that's the DNA of strawberries. And the reason that we see so much in strawberries is that if we go back to the slides, the natural strawberry and the woodland strawberry, this little guy here, when we've sort of bred successive versions of strawberry, and this strawberry, which you buy in a supermarket, has 8 times the amount of genetic information that that strawberry has. And people would say this is natural breeding. I would call it genetic modification. And there's a huge amount of DNA in your strawberry. So every time you're having a lovely strawberry, just think of all of that wonderful DNA that you're eating. It's a huge amount. Okay, so now that's the tutorial over with. What is synthetic biology, and why did I bother spending a few minutes going through all that? Well, synthetic biology essentially aims to design and engineer biologically based parts, devices, and systems, as well as redesigning existing natural biological systems. And there are many, many reports on synthetic biology around the world. One of the really impressive visions, I think, of synthetic biology is this. It has a powerful vision for merging the engineering design practice into the systematic construction of biological systems at the genetic level. And so you've been holding up cards, you've been essentially acting like a gene sequence. This is part of the foundation and fundamental aspects of synthetic biology. So we're going to now take this genetic information and we're going to now apply engineering practice and systematically reconstruct new biological systems at the genetic level. This is a very influential report chaired by my co-director, Richard Kitney, who I think is in the audience. And Richard and myself spent a lot of time writing that. It's a very influential report. You can download it. And we also have a little primer book. Sorry to plug it, but if you want to know more about synthetic biology, please look at that. Now, why now? So this is a big key question. Why are we talking about synthetic biology now? Well, this is an amazing period of time. This is from 1953 right through to 1981, about a 30-year period. And in that 30-year period, we learned what the structure of DNA looked like. So we learned all those principles I told you about. We learned how to take DNA from one organism and transfer it into another organism. We then learned how to sequence the DNA, read the DNA. So those are the letters I showed you, GCAT, GCAT, in 1977. And then we all, in 1981, learned how to synthesize DNA, chemically synthesize it. These are actually fundamental aspects of synthetic biology. And of course, 60 years later, we have the genome sequences for pretty much every major organism on the planet, including this one, which is the sort of one of our ancient human beings, which I think is an extraordinary genome sequence. So you can tell that this is a complete revolution in knowledge base for human beings, that we now understand essentially the instruction codes for pretty much every living organism. That doesn't mean we know how those instructions work, but we do know what the basic code is for many, many, many different organisms. One of the big drivers there, of course, is the ability to read and write DNA. Now, this is the price shown here in dollars of reading DNA. And you can see in the last sort of 10, 15 years, it has shot down. It is incredibly cheap to read DNA now, such that you can get your own genome sequence if you want. Now, the writing DNA is shown in the red and the yellow, and that's coming down. And we believe that there are some transformative technologies which are going to take that right down so that writing DNA, potentially in the next 5 or 10 years, will be as cheap as reading DNA. That opens up a very different opportunity. Governments have recognized it. So here's the World Economic Forum. They suggested that synthetic biology and metabolic engineering is the second most emerging technology with the greatest potential to provide solutions to the most compelling social, economic, and environmental challenges. That's a really big statement. Richard Kidney and myself were involved in the Synthetic Biology UK roadmap, and Richard's been involved in the refresh of that roadmap, so you can download that. So the UK has a complete governmental kind of position on synthetic biology. And I think one of the drivers, of course, is that it's estimated that the global market for synthetic biology could be 12 billion by 2018, although those are very difficult numbers to predict, but it's a huge number. So in simple terms, synthetic biology considers DNA as a programmable language. So we're considering this genetic code as programmable, such that we can sit in our computers, use our knowledge base, and build and design new biological cells, new biological systems, new biological functions. But how do we program cell at the genetic level? How do we do that using synthetic biology? So we then introduce this very, very powerful paradigm of engineering design practice. And I'm going to explain a little bit about why this is so powerful. Now, if you look at engineered systems, there are sort of 4 main aspects of complex engineered systems: systems control, redundancy, modular design, and structural stability. Those are really key aspects of the most complicated engineering systems around that we've made. So, I have enormous respect for engineering, because you can see the extraordinary things that engineers have done around the world. Now, if we start thinking about biology, can we start thinking about how we translate some of those engineering principles into biological systems at the genetic level? So systems control. So we have in cells feedback, feed forward. We have regulatory systems that allow cells to regulate themselves. Gene duplication, multiple metabolic pathways. We have redundancy in biological systems. We have modular design. We have evolutionarily robust multifunctional units that are shared amongst different species. We have structural stability. We have stable gene regulation, metabolism, and signaling. So we do have the kind of conceptual equivalence, if you like, around engineering complex systems that you need to engineer complex systems. So can we build biology with biology? And that's what synthetic biology is trying to do. So biological systems are modular. I've said that. The function is encoded in DNA. We have this huge knowledge base of genome sequences. I showed you that. Large diversity of genetic parts, which we know the function of, increased understanding of molecular cell biology, and a massive array of new technologies and techniques that allows us to probe living systems. However, big however, okay, biological systems are complex. They're multi-scale and they're stochastic or random. And you can see on this slide, we've got a metabolic pathway, metabolic pathways for a simple bacterial cell, very, very complicated. We've got compartmentalization, we've got protein structures at atomic resolution, we have cells that are firing genes at different times, and we have this sort of little fact about life, time. Living systems live, so they have a dynamic component to them. So designing and constructing living matter poses huge challenges, really big challenges. Biology is not plug and play. It's context dependent. Evolution, adaptation, natural selection, non-predictive stochastic behavior. That's how you get evolution. Emergent behavior that was not predicted. Self-assembly, emergent properties of biological systems. Non-linear dynamics, multi-scale interactions, because biology works at many different scales, as I showed you. And living cells essentially are constrained volumes of biochemical components. So this is a very big challenge when you're thinking about design. So how do we challenge that? How do we take that challenge on? Well, this is where engineering comes to play. We start using systematic design. So this is the big part of synthetic biology that is completely novel and completely unique. So what do I mean by systematic design? And I know Richard's in the audience, but please, be kind on me. engineering has some really strong aspects to how it carries out design. One aspect, and I don't want to spend too long on this, is this idea of abstraction hierarchy. Now here we've got the DNA code. That is the parts, okay? Then we want to build those parts as modular units into another higher level module, and that's a protein production module. And then we want to make devices and make systems. So this idea of abstraction hierarchy allows us to design very, very complex systems at different levels. And the future, and this is how engineering kind of goes, is if you're working at the systems level, you don't really need to understand all the AT, because you have a big hierarchical abstraction to work with in the sense that parts or functions are known. Now that is a really exciting concept. So what do I mean? So what is a biological part? So in synthetic biology, a biological part is a piece of DNA. So we have DNA parts that come together, and you can put them into a plasmid or into the genome, and you can design particular parts together to give different functionalities. Now, this is a major part of synthetic biology. This is called the synthetic biology design cycle. It's slightly complicated, but I'll just quickly run through it. This is where you're thinking about, what do I want to build? What are the specifications? I want a cell that will detect an analyte or some pathogen. We're going to show you that later. I want to look about the parts. What parts do I need to bring together? What bit of biology do I need to re-engineer to give that function? How do I build it? I need to build these parts. How do I test it? And oh, it doesn't work. Oh, the cell lost something happened. Okay, what happened? I learned. Let's go around. Oh, I've got to change the design, tweak it a little bit, change that there. And you go around this huge cycle, and suddenly you're into systematic design, and you're really beginning to constrain the complexity of biological systems. And this is a very important part of synthetic biology. So a workflow in synthetic biology might be design at genetic parts, do some modeling, build it, and test it. This is the classic engineering design cycle. So where do we get the parts from? Where are all these parts coming from? Well, this is where the genome writing comes from, the genetic writing, the DNA synthesis. So here we've got a whole bunch of companies that are now producing synthetic DNA chemically that you can order over the internet. So I can sit on my computer, I can design a genetic circuit, hit return, it will go out to one of these companies. I have to be a part of a proper regulated institution, I should hasten to add. I don't think anyone can do this, thank goodness. But and then I can get the parts that I've ordered back into my lab, and then I can start constructing and testing. And there are a lot of different companies now doing this. So what do I mean by standardized parts? So the idea of engineering is you have standardized modular parts. So here is a nice genetic circuit, and this one actually is very relevant to some of the experiments later. And these are sort of standardized parts. So that's a promoter, that's a ribosome binding sequence, that's a protein open reading frame. That would be like, I showed you earlier the HIV protease. That's a terminator. Stop transcribing the code at this point. Start transcribing the code at that point. So that's a promoter, a ribosome binding sequence, and that's a fluorescent protein and a terminator. So I think you're getting the point about genetic components, genetic bioparts that you can start building and designing. Now, Dick and I are co-directors of the UK Innovation Knowledge Centre called SymbiCity for the translation of synthetic biology into industry and into commercialization. And we built this automatic characterization platform. So here is a robot that is now beginning to carry out some of those processes of the design cycle. And this robot will be looking at multiple parts, and it will be analyzing them. This is in microbial cells, and this is fully automated. So this robot will be able to essentially characterize 1,000 parts a week. And obviously it's scalable. And we're then moving on to mammalian cells and other cells. But it's a very systematic process, and all of the information leads to these kinds of data sheets, which engineers are very used to developing. And I think the point about data sheets, so that's a little bit of DNA sequence as a part. It's a promoter. You can look at all the different characteristics of it, how it works, how you measured it, all the rest of it. And then this goes into Dick's SYMBIS, Synthetic Biology Information System, which Dick is building at Imperial College. And here we've got the parts all registered, all stored, all the metadata, all the integration of the knowledge is through the Synthetic Biology Information System. And so this kind of concept then allows you to design, and I think that's one of the key aspects. And this foundational technology, design tools, DNA synthesis assembly, parts, devices, characterization, looking at cells, how do the cells work, how do they work with my bit of genetic component in them, testing and prototyping. This foundational technology has this extraordinary ability to feed into many different application areas, so diagnostics, therapeutics, crop soils, fine chemicals, bioremediation, bioenergy, biomaterials. So the vision, if you like, for synthetic biology that certainly that Dick and I have been pursuing for the last 8 years or so, or more actually, is foundational technology is the key to the future of synthetic biology, and then all of these different applications can actually be tackled. Now, some healthcare applications. So I better check on my time. So I thought I'd better do some healthcare applications because I am in the Royal Society of Medicine, and I thought it might go down well if I didn't choose things that were not relevant. This is a fun project. This is a student project, 2009, from the Cambridge iGEM team. So here it is. What if you were to take a lovely yogurt drink, but you had all these engineered bacteria, and in your stomach, these bacteria were sensing all sorts of different ailments in your tummy, colorectal cancer, colitis, rotavirus, and changing color depending on what biomarker they detected. And then every morning you could go to the toilet, have a look at your feces, see what color it is, and work out how well you are. And I think this has got legs. I think this is a great idea. And they called it eChromI, cheap personalized disease monitoring from the inside out. Great project. And I was thinking, well, this is fantastic. Okay, here we are, 2014, 2015. Programmable bacteria detect and record an environmental signal in the mammalian gut. So the first part of that project has already been achieved. Programmable probiotics for the detection of cancer in urine. So, this is now beginning to be not just a fanciful story. Oh, isn't it fun to do that? What about this? This is now serious research going on about programming probiotic bacteria to detect, sense, respond, and deliver outputs that we can understand. What about engineered bacteria to detect and kill cancer cells? So again, the concept of engineered E. coli, maybe in your colon, detecting colorectal cancer, delivering some kind of signal to that cancer cell, and then that cancer cell being stressed or going into apoptosis or whatever. Now, this is a very nice work. So you think, well, is that happening? Okay, so the first happening was in 2006, where Chris Voigt's lab decided they engineered bacteria that could recognize cancer cells through the hypoxic environment of those cancer cells. And then this paper came out recently, programming human commensal bacterium to sense and respond to stimuli in the murine gut. So these things are happening now. Engineered phage bacteria, so these are bacteriophage, they're natural killers of bacteria, so why don't we engineer them so that they'll target pathogenic bacteria? Here we are, engineered bacteriophage targeting gene networks as adjuvants for antibiotic therapy. Engineering microbes to sense and eradicate Pseudomonas, Aeruginosa, a human pathogen. Engineered phagemids for non-lytic targeted antibacterial therapies. This is now becoming a very serious potential application space for these kinds of technologies. Now, engineering T cells has been around a while, but the T cell community have got very excited by synthetic biology, such that they're writing review after review after review. Adoptive T cell transfer for cancer immunotherapy in the era of synthetic biology. So there's an era. So we're moving away from traditional molecular biology into proper systematic engineering and design. T cell engineering as therapy for cancer and HIV, our synthetic future. Thoroughly recommend those that are interested to go and have a look at some of these papers. Now, finally, this systematic cellular reprogramming stem cells has been around a long time. The idea of taking adult fibroblasts and then somehow inducing a series of transcription factors and essentially reprogramming those cells to become pluripotent stem cells. And then, of course, once you have pluripotent stem cells, you can start differentiating those into many different tissues and organs that you might need to repair in terms of regenerative medicine. Now, this has been around a bit, but now you're getting really interesting papers coming through the literature where they're doing, highly efficient reprogramming, synthetic modified messenger RNA, synthetic biology mammalian cells, next generation research tools and therapeutics. This paper just came out last week, I think, Cas9 gRNA engineering for genome editing, activation and repression, and synthetic biology, rational pathway design for regenerative medicine. So a huge amount of interest in applying some of these technologies, systematic technologies, into stem cell research. Now, what about biosensors? So that's something we're going to show in a minute. So we've been working on biosensors for many years. And one of the visions, and if you like, one of my dreams would be to have a simple sort of filter paper test which I could dip into some sample and I would know whether it's a virus or a bacteria or a parasite just through a very simple colorimetric test. And we've been working on that for some time, but so has other great people. As you say, no idea is novel anymore. And Jim Collins published this paper a little while ago about paper-based synthetic gene networks. So this is complicated genetic networks which are actually incorporated into filter paper, which will provide some kind of biosensing signal. And we're going to show that in a minute. And then there's other papers coming out on biosensors and then some of our own work as well. So I think biosensing is a huge area. Now, the last project I want to describe, I think, is an amazing project, and this is to do with malaria. I don't need to tell people that malaria is a huge problem in the world. Billions of people at risk, many people infected, and of course, an enormous number of deaths. Now, this is a person who won the Nobel Prize last week, 2 weeks ago, maybe 3 weeks ago, who's a Chinese scientist called Yu Tu. She discovered a molecule called Artemisinin, and Artemisinin was found as a natural product in the sweet wormwood plant, which grows very well in China. So she spent her whole career identifying and isolating this incredibly powerful anti-malaria, driven actually by, I believe, by the Vietnam War era. That molecule is now a major way of treating malaria. But it's grown as a natural product and harvested. Now here's Jay Keesing, an amazing American scientist who decided, well, why do we, let's try and make Artemisinin in a yeast cell. Let's take the genes that make the molecule from the wormwood plant. Let's transfer them into yeast. Let's systematically engineer a metabolic pathway to produce the precursors of Artemisinin. And guess what? He achieved it. And that was the first publication in 2006. A partnership between Amyris, Bill Melinda Gates, One World Health, and Sanofi have essentially scaled that process up and are now producing Artemisinin tablets. And this is the latest from Jay, who sent me this last week. So they got approval in 2013, 35 tons produced 2013, 50, 60 tons 2014, which is enough for 100 to 120 million people. 2014, they did first 1.7 million treatments, and now May 2015, this year, 15 million treatments so far shipped to Africa from this particular process. It's an incredible achievement, but it did take 10 years to achieve. Right, so that's now on to the next part of the talk, which I'm hoping will work. So we're going to show you some of the biosensing technologies that we've been working on. I'm going to introduce that a little bit now. So in vitro diagnostic biosensors, massively important area. I think everyone who's connected with medical healthcare will know that. There is a great deal of sort of government healthcare savings, personalized medicine, point-of-care diagnostics, a great movement to have tests that are at point of care. And I think there is still a continuing need for cheap, reliable, simple biosensing systems. So my question is, could synthetic biology provide a next generation of biosensors? And I think this is one of the things we've been working on in my lab for some time. So just to give you a bit of background, so what do I mean by biosensors? What do I mean by synthetic biology biosensor? So a synthetic biology biosensor is essentially, could be two things. It can be a living cell, a whole bacteria. It could detect a signal. You have engineered code into that bacteria, which is running. That code is designed to detect that signal and then trigger a color change. So upon the presence of a pollutant, a pathogen, or some chemical signal, the cell turns red. And this is designed genetically encoded sensing modules in bacterial cells. Now, that's a living cell. Now, there is another type of sensing device which we're working, which is called cell-free. Now, what do I mean by cell-free? What I mean by cell-free is take a living bacterial cell and then kill it, lyse all of the good stuff out of the cell, not the genetic information, put it in a test tube, and essentially what you have is a biochemical cell extract which you can run your DNA programs in. And the programs will run very well in that because all the machinery for the translation that I showed you earlier in the transcription are contained within this biochemical extract. You do need to add some energy and a few other things in, but it's a very powerful platform. So we're going to demonstrate two novel unpublished DNA-encoded biosensors for medically relevant targets. So Kei-An Wen and Dr. Alex Webb are going to do that, hopefully. And the first project I think is very relevant, is the detection of Pseudomonas aeruginosa in cystic fibrosis patients. We're using synthetic biology designed biosensors, and the motivation for this was talking to Jane Davies, a colleague at the Brompton, about the testing of sputum samples, which is what they normally get, for Pseudomonas infections. So as you probably know, microbial biofilms is a big problem, commonly found in indwelling medical devices, catheters, wound infections, and obviously in a cystic fibrosis lung, there's a huge problem with Pseudomonas biofilms. And 65% of hospital-acquired infections are biofilm-based, so there's a great deal of need to try and detect this kind of thing. So what creates biofilms? What makes them form? So clearly, the bacteria, which are normally freely swimming, called platonic, they colonize surfaces, and they kind of induce this kind of signaling cascade, and they produce these small molecules called autoinducers. So these little molecules diffuse out, other bacteria sense them and say, hey, that looks like a good place to go and colonize. And they have this incredible cascade of signaling. The bacteria then form this biofilm, which is an incredibly resistant material, but also incredibly difficult to treat. So we thought, let's try and think about how we would tackle this particular problem. Now, obviously in cystic fibrosis patients, they suffer terribly from persistent Pseudomonas infections. And in talking to Jane, we wanted to develop a very cheap, simple synthetic biology biosensor that could be used in a clinic or even used at home to detect infections. But we also wanted to think about the physiology of the organisms, the physiology of the bacteria within the cystic fibrosis lung. And so we chose these Quorum sensing molecules. So these are the little molecules that bacteria use to signal for biofilm. Pseudomonas has two specific molecules called 3-oxo-C12, HSL, and C4. These are very specific for Pseudomonas aeruginosa. And Burkholderia uses another molecule. You'll notice these molecules are very similar. They're very similar structures, but they are distinctly different. So what Kayan has done is that she's designed a biosensor that is taking a bit of Pseudomonas biology. This is the machinery that Pseudomonas uses to detect that little signal. And she's refactored it or re-engineered it or redesigned it into a very simple biosensor. And so it's based upon this protein, which recognizes specifically this small molecule. Only when it has that small molecule will the protein come together and form a dimer, shown here. And only when it's in that form will it bind to another DNA sequence and activate the transcription of an output signal. And we've engineered it to be GFP. Now, this is the code. This is the device. This is the device that she's designed and engineered. And I just wanted to illustrate, this is a single large piece of DNA from modular parts, different systems put together. And I'm going to ask Kayan to come up and show that in a minute. You can come up now if you like. Sputum samples, we've worked out a way of trying to deal with these samples for this particular test. And this is the great results. This is the first time, this is a bit of data, sorry about this, but I couldn't resist showing it. So this is, I think, the first time that a synthetic biology biosensor designed and engineered has been used to detect a particular analyte in a patient material. And this is unpublished, and we're really excited by this breakthrough that Kayan's done. But for the purposes of this talk, come up and show us some of the results that you've done. So what Kayan's done is that she's taken that design that I showed you, which is essentially a piece of DNA, but it's refactored a bit of Pseudomonas biology to detect this little small molecule that Pseudomonas uses to communicate and to signal. And we're going to, and she's did this experiment. This is a cell-free experiment, so don't worry. There's no Pseudomonas aeruginosa in anywhere near here. There's no actually. This is fine. This is all fine. This is a cell-free extract that Kayan's done. And what she's going to show you when this works in a minute, lights will go down. You'll see two tubes there. You can see the two tubes. Now you'll see one tube shown here is green fluorescence. And that is the output from the biosensing device that is specifically engineered to detect the little small molecule homoserolactone that only Pseudomonas aeruginosa pushes out. And so in that experiment, she took a cell-free extract, she took the DNA, she put the DNA in it, she ran it for 30, 40 minutes, however long I've been talking, and then she added the analyte, that's the small molecule that is meant to detect, and you can see quite clearly there that it's gone fluorescent. Now, obviously, that as a point-of-care diagnostic is not, we've got a blue light box here, that's not exactly a very simple system. But as a proof of principle, it's a really clear example of the high specificity that you can engineer into biology. Now, on that other, now what Kayan's got there is some other bacteria. This is in a sealed plate. This is just lab strain E. coli. It's perfectly safe. And what we've shown there is we've taken some output proteins that you might use. So this is a green fluorescent protein from jellyfish. And this is the red fluorescent proteins that are one's from a coral and another's from a jellyfish. So this concept of taking genes from nature, taking proteins, taking structures, putting them into the context that you want to do. And these modules here can show, this kind of shows you the modularity of synthetic biology. So we can change our output module, and we might put a red module there, or we might put a blue module. So thank you very much. That's great. I'm glad that worked, actually. Anyway, great. So that's just to show you a little bit. Okay, so that's a proof of principle. It's not a device, but we're working very hard to think about how we might make that device. And, as I said to you, the dream is to have it incorporated into one of these strips like this and just dip it in, and that's what we're hoping to achieve. The next project is a global health biosensor project. Again, unpublished data, but it was so compelling I couldn't resist telling you this story, actually. And this is a fantastic story, and Alex Webb's been working very hard on this. And this is about trying to detect schistosomiasis parasites using synthetic biology designed biosensors. And it's a great collaboration with Joanne Webster and the Schistosomiasis Control Initiative, and also the Upstream Alliance we're working closely with in terms of doing some field tests on this device. So, just to introduce it, you probably know it's a horrendous parasitic disease. Again, it's millions of people suffer from this disease. It's also known as Bilharzia. The symptoms are chronic disease, and it's very debilitating for those societies. There is a primary human infecting species called Schistosomiasis mansoni, and there are many other species that actually affect cattle. So in these villages and environments, not only is people getting infected, but actually livestock are getting infected as well. And it's a very difficult disease. Now, as you can probably guess, it's very, very widespread. There are different types and versions of this disease, but you can see the continent of Africa has a significant burden of this disease, as does China and parts of South America and also Southeast Asia. Now, the really cool bit of biology about this parasite, and it is an incredibly evolved beast, is that it has this extraordinarily complicated life cycle. But the bit I want to focus on is this bit here, which is the infective part. This parasite uses us as a host as part of its living life cycle. And so it gets shed into water. It's actually living very happily and maturing within freshwater snails that exist in water courses. And then at some point, these parasites, the Siccaria larvae, get shed into water, and then that larvae is somehow attracted to human beings, and particularly to the skin, and particularly to various fatty acids that we have in our skin. And then that's the snails, that's the larvae, and of course in various water courses you can imagine how easy it might be to get infected by this terrible parasite. And hematuria is a classic symptom of infection. So here's the infectious stage. This little larvae is seeking out human hosts and also animal hosts as well, hits the skin, and upon hitting the skin, it releases a massive amount of enzymes that essentially dissolve away the elastin of your skin and allows it to bury its way into your body. And we were very interested in that aspect of the infection. We really wanted to know whether we could detect the active infective Siccaria larvae. Now, the current detection systems are a bit ridiculous, I think, because they are, this one is extraordinary. You infect mice and check back after 6 weeks for infection. I don't see that as a very quick diagnostic test. There are other tests which are actually even more cruder. Look at human stools for eggs. I don't think that's very helpful. Now, then there's more sophisticated tests like PCR or immunoassays. But of course, where we're talking about some of these assays and this type of research tools will be extremely difficult. So this is what people do when they go and look to see where these infections are. They go out, they collect the snails, they bring them back to these field laboratories, they look underwater, and they look at these tiny little Siccaria larvae, and they count them. And we just felt there must be a slightly more sophisticated way of doing this. And so what Alex and done, this was inspired by a student competition, actually, an iGEM team at Imperial College called Parasite many years ago. But what Alex has done is taken that on, and he's really pushed the project. So it's a simple biosensor. This time it's a whole cell biosensor. So what Alex has done is designed a protein that goes into the cell wall, and it displays another bit of a protein here, which is very, very specific for the elastase protease, the particular parasitic protease. So when you detect the elastase, that gets cleaved. This dye-bound molecule leaves the cell, and the cells become colorless. That's the theory, anyway. Here is the device. It's a very nice genetically encoded device. It's been designed specifically to do this. So if you imagine you have a red cell, this is E. coli, and in the presence specifically of this protease, it knocks off all these dye molecules, and the cell becomes colorless. Now, Alex, there's a device. So again, DNA programmability, that's what I wanted to get to. This is a bit of DNA. It's been designed, put together in different ways. Never exists like this in nature. This is specifically purpose-built, designed biosensor for the elastase protease. And Alex, I think you're going to come and show us a little bit about how it works. Now, yeah, and I think that's over here. So what Alex did early on is he ran a few DNA. I'll describe it when we show them. Ran a few experiments just at the beginning to show some of the outputs that we can get from this device, but he also did an experiment before we came to show you the outputs that would exist. Now this is great. I mean, I just love this experiment. So here we've got, these are little bacterial cells in the bottom of the tube there, and that's the negative control. You know, always have negative controls. If you're studying to be a researcher, key thing, you must have a negative control. Now here's a positive control here, and that means that all of the dye, if you like, is on the surface of the cells, and you can see that there. So that's perfectly expressed, properly, nicely displayed, if you like. I can't read your writing. What does that say?
So that it's not recognised the protease. So
right.
It's not
so in this tube, I think Alex has put in the protease, which is not designed to recognise, and therefore the cells still remain red. And in this tube, he's put in the protease, and obviously that's a purified protein, into the tube, and all the cells have essentially gone completely colourless. And this is a very beautiful example of a very simple conceptual design and working highly specifically, which I think is fantastic. Then Alex has also done some experiments where where he's ran some DNA codes in cell-free extracts, so not living cells, and he's managed to show some outputs here. So again, a negative control, and this would be a more colorimetric output. When I say colorimetric, it would be a very defined color. You know, it would only happen when you have the elastase present or the protease present. And there you can begin to see the different kinds of strong outputs. And this would be leading into a design that would allow us to have a positive signal. And that's worked quite nicely, I think. Great. Okay, brilliant. Thanks, Alex. Okay, I probably need to, oh gosh. Okay, time, gosh, yeah. So how would we implement this design? So one of the students for the Parasite project came with this beautiful idea of having a little water device here and encapsulating linoleic acid, which is the fatty acid that would be in your skin. Then taking the water, all the Saccharella larvae would swim towards this, release their enzyme, and then we would detect the color change within that device. And that's something we're exploring with Joanne and colleagues that are working in the field on this. In fact, these guys, the Upstream Alliance, who have got this amazing project where they're putting a nematode into fresh water, which actually feeds off the snails, and they're trying to introduce this nematode and see what consequences it have on clearing infection sites. And they're desperately keen to have a biosensor that would allow them to know whether there is any active Saccharella larvae in their samples. Okay, now we should stop at this point, I think, because I've gone on about all this fantastic stuff with synthetic biology. We're gonna do huge numbers of interventions, therapeutic interventions, biosensors. We're going to make drugs that exist in natural products. We're going to do biosensors that are going to go out into the field. We're going to solve global health problems. We're going to do it. But, okay, big but. Okay, this is what people think of synthetic biology. Some people think of synthetic biology. Maybe you can have a read from these in the back, but I can quickly read them. Okay, scientists accused of playing God after creating artificial life by making designer microbe from scratch. But could it wipe out humanity? Probably not. But anyway, Mail Online. Here we've got a good one here. I don't know if anyone's familiar with the American TV series called, I can't remember what it's called, but anyway, Brewing Bad, scientist finds way to cook up heroin at home. This was after a beautiful paper was published showing you could make opiates in yeast. Again, along the lines of Artemisinin, because we're completely dependent on poppy supplies, and therefore it might be better if we could think about other ways of making opiates. Here's another one, which is stop, what does this one say? It says, put a stop to synthetic biology pollution. So there we're talking about, and then this one here, synthetic biology, and the next assault on biodiversity. And this one here, Ecova and method, now with extreme genetic engineering. And this is my favorite of all of them. It's a beautiful image. So this is a beautiful piece of ice cream with a horrible laboratory pipette, extreme genetic engineering in your ice cream. It's a very evocative image. Tell, Haagen-Dazs, and Baskin-Robbins, and etc. reject synthetic biology vanilla. Kickstarter bans GMOs. Do we really want to revive the woolly mammoth? Probably not. But you can see there is a lot of opinion about synthetic biology outside of the scientific circles. And I think quite rightly so. I think this is a technology that has very powerful ramifications. Now, to try and address that to some extent, there is something which has been developed called the Responsible Research and Innovation Agenda, particularly led out of the UK and also in Europe. And the idea here is that we're trying to build about some sort of creating value for society in an ethical and responsible way. So it's about being responsive and potentially adapting the research itself in order to develop a technology that is more socially desirable. It's not about changing public opinion. And this agenda is very much embedded in Dick and I's research center and in everything we do. And we work closely with social science in King's College. And in fact, all the synthetic biology community have extremely strong links with social scientists, which is a very interesting interdisciplinary collaboration. My view, I think, is here. The limits of synthetic biology, I think, will not be technical, but societal, in that the applications and utility will need to be proven and accepted. And I think that is probably where we're at the moment. I don't think there will be technical limitations placed on this technology. I think it's very, very powerful.